All right, so glycolysis is a process to generate ATP. So it's going to be tightly regulated because if you don't need ATP, you're going to have to do something else with the carbons in your glucose molecules. So again, we're going back to the other functions of glycolysis. So glycolysis is going to be able to take the carbons in glucose that it took up, and it's going to be able to pass them off into other pathways, including glycogen synthesis for storage, pentose phosphate pathway for the 5-carbon sugars. We're going to make fats if you're in the liver. You can make different amino acids with your carbons from glycolysis. So there's going to be other functions of glycolysis than just your making up your ATP. Now, glycolysis is going to be regulated by need for ATP. Again, if you have too much ATP, you're going to start to go into storage. And so glucose 6-phosphate is going to have its phosphate added by a hexokinase. And hexokinase is going to be negatively regulated by its product, glucose 6-phosphate. So this is feedback regulation. Now you also have fructose 6-phosphate, which will become fructose 1,6-bisphosphate. And this is our rate limiting step of glycolysis. Now the reason glucose 6-phosphate is not the rate limiting step is because this has other fates. So it does not have to go through glycolysis. Now, once you have fructose 1,6-bisphosphate, it has to push through glycolysis, the other steps of glycolysis. And you can see here that fructose 1,6-bisphosphate is going to be made by phosphofructokinase 1, which is called PFK1. And this is going to be regulated. This is going to have allosteric regulation. By many compounds. So it's going to be positively regulated by AMP, which makes sense. So if you have a lot of AMP, you want to make ATP, and you want to push your carbons through glycolysis. It's also going to be negatively regulated by ATP, which makes sense, because if you have too much ATP, you're not going to want to make any more ATP, and so you want to turn off the push of carbons through glycolysis. Now there are other regulators as well. There is citrate. Citrate is part of the TCA cycle that can come into the cytosol if it accumulates, and it can negatively regulate glycolysis. So citrate is from the TCA cycle. Fructose 2,6-bisphosphate sounds a lot like fructose 1,6-bisphosphate, and it is. So fructose, if you have an accumulation of fructose, or if you have fructose 2,6-bisphosphate, it's going to be a positive regulator of pushing towards fructose 1,6-bisphosphate. We'll talk about that. So fructose 2,6-bisphosphate regulation of glycolysis. So again, let's see what's going on here. Now first we have glucose on this end, and we have our triose sugars over here. You have your DHAP and your glyceraldehyde 3-phosphate. In addition, you have your fructose 1,6-bisphosphate. Remember, this is the guy that's the rate limiting step. So you're going to have your fructose 2,6-bisphosphate regulate the formation of fructose 1,6-bisphosphate. Fructose 2,6-bisphosphate is generated directly from fructose 6-phosphate. What this means is that if you have an increase in your fructose 6-phosphate, you are going to increase fructose 2,6-bisphosphate. But your body can't do anything with fructose 2,6-bisphosphate. It cannot put this through glycolysis. It's sort of like this is a holding place for the carbons because there's too many carbons, and so it's going to accumulate in this form. When this form accumulates, it wants to turn on PFK1 to make fructose 1,6-bisphosphate because the carbons are stuck in 2,6-bisphosphate forms. And so it turns on the enzyme that's going to allow for fructose 6-phosphate instead of going to 2,6 become 1,6. And it's only the 1,6 that can be pushed through glycolysis. Now again, our PFK1 is our rate limiting step. This is going to be positively regulated by AMP like we just talked about, positively regulated by too many carbons stuck in fructose 2,6-bisphosphate form. It's going to be negatively regulated by ATP and negatively regulated by citrate. Now on the other side of this, over here, this is in the form of gluconeogenesis. And the liver has a fructose 1,6-bisphosphatase. And this is going to remove the phosphate on that one position to regenerate fructose 6-phosphate because in the fasting stage, the liver wants to make glucose. In the fed stage, you're going to push the glucose through glycolysis. Now you can imagine that this is also going to be negatively regulated by need. So if the liver has low ATP levels, it needs to make ATP in order to release glucose. Okay, so apart from this, you can see here that we have two enzymes that are involved in the formation of fructose 2,6-bisphosphate or its breakdown into fructose 6-phosphate. And it's the same story. We have a kinase that's going to add a phosphate, and in this case, it's phosphofructokinase 2, not phosphofructokinase 1. And phosphofructokinase 2 adds a phosphate to that 2 position. And phosphofructokinase 1 adds a phosphate to that 1 position. And then when we want to remove a phosphate from that 2 position, we need phosphofructokinase 2 phosphatase. And that will remove that 2 position. When we want to remove the phosphate from the 1 position, we need phosphofructokinase 1,6 to remove that 1 phosphate. And that's how it's regulated with respect to fructose 2,6-bisphosphate. Now another step that's going to be regulated is our pyruvate kinase. So our step here, that's going to take PEP to pyruvate by our pyruvate kinase is going to be regulated. Now again, think about what we just talked about. Fructose 1,6-bisphosphate is up here pushing through glycolysis. So this is, in essence, a feed forward. Regulation. Fructose 1,6-bisphosphate is saying, I'm here, you need to keep pyruvate kinase on because we need to push these carbons through. Now, of course, negatively regulating this is going to be ATP, because if you have too much ATP, you don't want to generate ATP. And remember that there are some options for your carbons if it does go down the route of fructose 1,6-bisphosphate. You can still make, for example, serine or other amino acids. So you still have options at this place if you don't want to push through to make ATP. Now another regulated step is pyruvate dehydrogenase. Now this is going to be in the mitochondria. So we'll introduce it here. We'll talk about it more when we talk about TCA cycle. But again, if you need energy, and in this case, you're going to be regulated by your ADP, and calcium. Calcium release from the sarcoplasmic reticulum will activate the mobilization of energy. And so it's going to turn on any of these enzymes that are going to potentially push the carbons through to generate ATP. And one of these is pyruvate dehydrogenase. Now it's going to be negatively regulated by NADH. So again, this is our feedback. So this is feedback regulation. If there's too much NADH, it does not want to make more NADH because you don't have enough NAD to pick up those electrons. It's also going to be feedback from its product. So if you have too much acetyl-CoA, you're not going to want to make any more acetyl-CoA. So that will negatively regulate pyruvate dehydrogenase. So phosphofructokinase 1 and pyruvate dehydrogenase are going to link glycolysis and the TCA cycle. And both are major regulatory sites. And so again, you can see that we have regulators of AMP, we have regulators of ATP, and we have regulators of ADP. Now there's a reason why all of them are used. ATP obviously to shut down, making more ATP, so this usually turns off. And then these two will usually turn on. Now the body is smart because it doesn't make all of your enzymes that need to be turned on sensitive to ADP or AMP. It kind of mixes it up to make sure that it kind of covers all bases. Because sometimes you might have an increase in ADP, or sometimes you might have an increase in AMP. And so different enzymes are going to be regulated by different versions of that molecule for those reasons. So ATP, ADP, and AMP have certain levels in the cell. And levels of AMP in the cytosol are a good indicator of ATP utilization because AMP and ATP can make 2 ADPs. And really what it comes down to is what can you detect. So if you're going from a 4 to a 5, this still looks like you have quite a bit of ATP. If you're going from your 1.5 to 1, you still have a good amount of ADP. However, in AMP's case, you go almost to 0. So that's easier for your cells to recognize as being too low, is when you have no AMP left. And so this is going to help to regulate and turn on PFK1 so that you can push the carbons through glycolysis. So adenylate kinase is going to hydrolyze ATP to ADP and it's on an increased or break down AMP, ATP back to ADP, etc. All right, so PFK1, let's go back and visit this one. Remember, we started with glucose, we added a phosphate to that 6th position, we changed it to fructose, and then we added a phosphate to the 1 position. We made fructose 1,6-bisphosphate. And this is from phosphofructokinase 1. Phosphofructokinase 1 has 6 binding sites. It's a very large enzyme and it has a lot of interactions. 2 are going to be substrates, ATP to transfer that phosphate, and fructose 6-phosphate. And then the other 4 are allosteric regulatory sites. So you can see here on our diagrams here is fructose 6-phosphate levels are going to be impacted, or the need for energy are going to impact the conversion of fructose 6-phosphate to fructose 2,6-bisphosphate, which is a positive regulator of phosphofructokinase 1 to make the 1,6-bisphosphate. And then ATP also, as it drops, you're going to increase AMP, and this is also going to drive the production of fructose 1,6-bisphosphate. So the regulatory sites are going to be inhibitory or activating. Inhibitory are ATP, or citrate, something else that is accumulating in response to too much energy. And activation is going to be those that accumulated with a need to push through glycolysis. It can be that fructose 2,6-bisphosphate, and it can also be things like AMP. Now, for phosphoproteokinase 1, it is an isozyme. There are different versions of it. Muscle is going to be very sensitive to AMP ATP. Liver is also going to be sensitive to AMP ATP, and the version C is going to be found in other tissues. Now, your tissues can have more than one of the phosphoproteokinase 1. Red blood cells are going to have both M and L subunits because red blood cells only use glycolysis. They want to do a lot of glycolysis. Your active PFK1 is another tetramer. Now, pyruvate kinase also has different isoforms. The R form is in the red blood cells. The L form is going to be in the liver. And your pyruvate kinase in the liver has allosteric inhibition. Your pyruvate kinase in your red blood cell does not. So the R form is typically always going to be on because there's nothing else your red blood cell can do with the carbon if it goes all the way through glycolysis. Your muscles have other versions as well. M1 is going to be in the heart and brain, and this has no allosteric sites either. And so this one is going to be on as well. Your pyruvate dehydrogenase complex in your mitochondria. This is going to be, again, it's going to be activated by the need for energy, which can be a lack of ATP or increase in ADP, or it's going to be a calcium influx. So it's going to be need-based. So a question here is if you have adequate oxygen supplies, but an increase in AMP, what is going to happen here? If you have inadequate oxygen, basically you can't push things through your electron transport chain, and then you're going to have to take your carbons and your acetyl-CoA and push them back out. And this is basically going to go to oxaloacetate, which is going to go to malate, which is then going to go into the cytosol. And you can basically donate your carbons, you can bring them back to the cytosol for other reactions. Now, the Wartburg effect has to do with cancer cells. So in 1924, it was observed that cancer cells would continue to ferment glucose into lactic acid even if there was enough oxygen present. And so what was found, this is called the Wartburg effect, by the person who discovered this. And so what is known about cancer cells is that they are mutated. So in the case of nutrition, cancer mutations cause them to take up glucose in excess of what they actually need. And so you're going to follow the same rules with respect to the enzymes. And if you have too much ATP, you're going to have to push those carbons somewhere. And those are going to go towards lactic acid in the case of many cancers. So lactic acidosis is where you're going to have an excess of lactic acid in the blood. And the threshold for excess is about 5 millimolars. Now, you're going to have, again, increase in NADH-NAD ratios, and this is going to direct pyruvate to lactate. So remember, here we are in glucose. We go through glycolysis and we generate pyruvate. We also generate NADH. So if you have an increased NADH-NAD ratio, that means you have too much NADH and too little free NAD. And so you're going to push your NADH through lactic dehydrogenase to form lactate. Remember, NADH cannot cross into the mitochondrial membrane. It has to use one of those shuttle systems. And if you overwhelm things, you're going to have to push those electrons through to lactate. Many causes of this are going to be excess alcohol, So alcohol is a toxin. It must be metabolized. And so if you rapidly oxidize alcohol, you're going to rapidly increase your NADH levels, and this could push things towards lactate. Another cause could be hypoxia, where you have a lack of oxygen, and that's going to push your things through to lactate and lactic acid. So basically, you have a decrease of oxidation of NAD and FAD, and this is going to push pyruvate to lactate. Or you have deficiencies or inhibition of your TCA cycle, and this is going to lead to an increase in pyruvate and lactate formation as well. You can have different poisonings, carbon monoxide poisoning, cyanide poisoning, and this is all going to target your electron transport chain, decouple your electron transport chain, and this is again going to push things to lactate. You can have some genetic defects in mitochondria DNA, so we're going to talk about different complexes, but if these defects don't allow you to metabolize the carbon coming into the mitochondria fast enough, those carbons are going to have to leave and they're going to go to the cytosol, and then you're going to form more lactate.